pleased to be here speaking with all of you today, and really excited to tell you a little bit about some of the work that our organization has been doing, but also just more generally to tell you a little bit about kelp. And we'll kind of span the range from I'll show you some nice photos. We'll talk about why you should care about kelp, and we'll take a brief foray into a few other things. But really the goal here is just to help folks learn more about why they should care about kelp forests, and what's happening with kelp forests in BC, and really what we as a group can do about it. So I'll start just by acknowledging the Kelp Rescue Initiative was founded 2 years ago with some very generous core funding from the Gampage family, and it's really helped us get started and get settled. And so in addition to all the other funding sources you see here down below, that is just a really important thing to start right off with. But enough of all that. I'll open with this image. So as a kid, I sometimes dreamed that I was flying above a forest, and I would wake up and there'd be that moment where I was disappointed that it was only a dream. And in lots of ways, growing up to be a marine scientist, I've discovered that diving, those kinds of places do exist, and they're called kelp forests. So diving into a kelp forest is as close to that dream of flying over a forest as I've ever been. There are these towering canopy kelps, so like this bull kelp that you see here, or giant kelp floating on the surface. There's understory, kind of like shrub kelps, all these understory species, 20 different species or so in the Salish Sea, depending on how you count. And in this forest, the flowers are replaced by sea anemones and sea cucumbers, catching prey from the water column, and these iridescent red algae. And that structure, just like a forest on land, creates little places for animals to hide, places for animals to feed, to lay their eggs. And these kelp are incredibly productive. They can grow up to 25 to 30 to even 40 centimeters a day for some species. And so they're capturing all this energy from the sun. They're turning this carbon dioxide from the water into sugars and into their tissues. And then the ends of their leaves or their blades are breaking off and kind of floating by, only to be caught by sea urchins that are crouching in all the shallows and all the little cracks and crevices, just ready to grab that kelp with their 2 feet and eat it. And so in these magical places, birds are replaced by fish, by octopus, which it sounds like you'll learn about later tonight, and they just really are truly remarkable and wonderful places. But they're also places with a bit of a reputation problem, because in lots of ways, most of us experience kelp forests not as a free diver or scuba diver might, but just as as folks on land, these slimy kind of brown things wash up on the beach. You might have used them to play jump rope once or twice or something, but really the kelp really needs some attention in terms of kind of their image, I guess. And so in some ways, part of what I want to do tonight is just try to convince you that kelp really are these magical, wonderful things, and what you kind of see at the end of their lives, often when they're washed up on the beach, certainly isn't the whole story. And any introduction to kelp forest wouldn't be complete without talking about salmon. So I've talked about some of these other species that depend on them. Well, salmon are really hugely tied to kelp and other nearshore ecosystems. So as these young salmon hatch out of rivers, they swim down into these estuaries where there's eelgrass beds and salt marshes, and they're kind of adapting to this new saltwater environment. They need these places to hide and to feed so that they can gain strength before they move on to their migration. And so you can think of kelp as just one of these really important stops along this salmon highway. So as these different Pacific salmon are leaving these estuaries, they're leaving the Fraser River, they head past these kelp forests, and they're really important habitats, both for them to get shelter, for them to get food, and for them to be protected on their way out to their open ocean migration, where they spend the rest of their, or most of the rest of their lives. And then they also use those adult salmon use those kelp forests when they're returning back to these rivers as well, both again as places to hide, to rest from the currents, and to feed on the many species that also depend on these environments. So they're hugely important for salmon. Now, this is a map that shows all of the world's coral reefs, and this is part of my effort to kind of raise the profile of kelp to this audience. So, one of the things you'll notice about coral reefs is globally is they're mostly centered on the tropics. They're a warm water species. They're a foundation species. We've all learned about their importance, and lots of us have probably learned about some of the threats facing coral reefs, including things like marine heat waves. And now this is the same map with the global distribution of kelp overlaid on top of it. And so it's really remarkable that when you think about it and you look across the globe, kelp forests cover 1/3 of the world's coastlines, which is an area 5 times as big as all coral reefs. So in terms of just the length of coastline where kelp forests are really this important primary foundation species, it vastly overshadows the same patterns for coral reefs. So they really are the corals of temperate ecosystems or temperate tertiary ecosystems. Yeah, and I guess I mentioned this, but just importantly, that kelp are a cold water species, and we'll come back to this later on in my talk as well. And kelp forests can put up with a lot. So this is an image, and I'll mention this is one of many images that I will show in my talk taken by an excellent wildlife photographer that lives near the Salish Sea named Fernando Lessa. If you haven't run across his work, you likely will soon. And this is an image of a kelp forest right in Burrard Inlet, traditional territory of the Tsleil-Waututh First Nation, among others. And, it's in amongst these sulfur piles and Canada's largest port and this shipping lanes and all these places, and yet it sort of persists. So there are certainly lots of places where, despite a lot of shoreline development and various other things happening, kelp forests do persist even in these urbanized places. So that it's sort of an exciting refuge in some ways. And it's really important for our work to understand why kelp are persisting in some of these places, so we can use that to help understand, what we can do to help make other places more amenable to kelp forests. Just another image, and back to the Salish Sea. So this is just a scan from the Comox District Free Press from October 1968. And, the headline really says it all: pesky kelp turned into useful fertilizer and other products. So really, if you think about, how we've been thinking about kelp forests for a long time, it wasn't to think of them as these really important foundation species. It was to think of them as this kind of pesky species to be turned into something more useful, right? But, to a large degree, kelp have tolerated some of this kind of harvest and some of this kind of disturbance. And really, I guess the other reason I'm showing this is that it really speaks to just how common kelp forests were in the Salish Sea, including in places like the North Salish Sea back in the 1960s and 70s. So really, they were so ubiquitous that there was these giant boats that were out there harvesting them for fertilizer. All right, so a quick digression. So I wasn't thinking about kelp forests back then. In fact, I wasn't even born back then. And when I was a scientist at University of Victoria years ago doing my undergrad and then eventually a PhD at University of Alberta, I studied species that depended on kelp forests, that live near kelp forests, and that included everything from crabs and invasive snails and how predators and prey interact, and also barnacles, which is why I'm putting up some of these headlines. So the work I was doing back then was really, even though all the species I studied were nearshore species, they depended on kelp forests, including these headlines about the sex lives of barnacles. I really took them for granted. So in lots of ways, this is just a picture of me in my much younger days and one of my field's collaborators working in a site near Bamfield collecting barnacles. And you'll see behind me this nice, healthy bull kelp forest. And we just took that for granted. We just assumed that those kelp forests would always be there. It was really the backdrop for much of my work in lots of ways, including literally in this photo, but it wasn't something that I paid much attention to. And then that all changed in 2016. So this was kind of the third year that this large marine heat wave, this large body of warm water had moved in out of the kind of central Pacific and had kind of run into our coast. And I'm sure folks on this call were quite aware of it. The blob, it was called. And so this patch of warm water moved on shore. It started in 2014. It moved on shore by 2015 and 2016 in our region. And it really warmed up the nearshore waters by quite a bit. And so during that time, I was teaching a field course out in Bamfield, the Marine Sciences Center, and we took our students out and we started looking at how the kelp forests were doing. We compared how the kelp forests were now in a bunch of those places to a historical data set collected in the same region back in the 1990s. And we were quite alarmed to see that we started we were seeing quite a few declines. Places that used to have kelp forests in the 1990s even no longer did after this marine heat wave. And so that really piqued our interest. And that's really what got me and my collaborators really started on looking at this. And at the same time, we were looking at this in BC. Folks were looking at the same pattern everywhere from Alaska to California, and I'll come back to that in a second. And the reason I'm showing you this image in particular, so this is it's a model output, but it's very similar to measured conditions in kind of August water temperatures in the Salish Sea during that blob event. And so you'll see a couple things. One thing, you'll see this big, anything red is warm, so you'll see this big patch of warm water really filling most of the Salish Sea, or certainly the central and north Salish Sea. You'll see this cool water kind of running in the Strait of Juan de Fuca and you know, spurred in part by these large current events that roll through there. And then you'll see in all these little inlets and fjords all the way down to Seattle and all the way up into a bunch of other places, including on the west coast of Vancouver Island here in Barkley Sound, these kind of small pockets of warm water that were in those places as well. And so what it turns out, as we've been studying that more, what was happening as that blob of warm water moved in was that it was, you know, kelp were really struggling to survive in that marine heat wave. But the other thing that was happening is that it brought with it or made worse a disease that was running through a whole bunch of species of sea stars, likely caused by a virus, something known as sea star wasting disease, that obliterated most of these large sunflower sea stars that used to be so common, and many other species of sea stars up and down the coast. And that's important because at the same time, those sunflower sea stars in particular, their favorite food is sea urchins. And so as the sunflower sea stars disappeared, 95% of them disappeared from Alaska to Baja California. That caused quite a large increase in the number of sea urchins that we see. So this is just an image on one of the local reefs, just showing what we now call, you know, an urchin barren. So a place where sea urchins have really mowed down all the vegetation, and all you have left is this large conglomeration of sea urchins. And if you look at aerial photos taken further north in BC, you can see that these urchin barrens are incredibly common in some places. And so this is an image, and it's probably a little bit hard to see depending on the size of your screen. So each one of these little black dots here, and then each one of these little this kind of red band here, right where the water meets the low intertidal zone, each one of those is a sea urchin. And so if we zoom in on that patch, for instance, you can see each one of these little red blobs is a sea urchin, each one of these black dots here in the water is a sea urchin. And so there's places in BC that used to have kelp forests that are now just blanketed by these sea urchin barrens. And that matters because you know, these forests without the trees, just like a clear cut on land, they're less biodiverse places, they're less productive places, they're not able to capture all that energy from the sunlight and turn that carbon into sugars for food and to provide habitat for these other species. And so it's a really, it's been a really challenging 10 years or so on this coast to see some of those changes happen. But so far, all I've showed you were some pretty pictures, and really that's in lots of ways in BC that's, you know, we didn't have a lot of detailed scientific evidence about how common some of those patterns were. We'd seen them in some pockets of the coast where we'd studied more broadly. We'd seen them in Barkley Sound, for instance, on the west coast of Vancouver Island. But how common were these patterns of kelp forest loss in the Salish Sea since the blob, as well as elsewhere in BC? So what me and my colleagues did is we compared some historical imagery taken by the province of BC called shore zone imagery, and compared that, some historical imagery from the 1990s or somewhere all the way up to 2007 to some more modern imagery taken between 2018 and 2021. And we looked at all these little sections of shoreline. So anywhere there's kind of a gray line along the shoreline is a section of shoreline we looked at, and we looked at each individual photo before and after this blob event, and we just said, hey, what's happened. And so if you add all those up for each region, you can count the number of photos and the number of sections of shoreline that have lost kelp, the number of sections that have been stable throughout that time period, and the number that have gained kelp. So basically the take home here is that anything red means a region or an area where we've studied where kelp forests are less abundant. There's been a loss between 2007 and about 2018. Anything that's gray means it's been quite stable, and anything that's blue means there's actually been an increase. And so what did we find? The first thing is that in northern BC, in the two places where we had data, there appear to be some substantial losses over that time period. And if we look at those images, there's lots of evidence from a lot of those images, like this one that I'm showing you here and that I showed you earlier, that sea urchins are certainly playing a large role in some of those declines. Whereas if we look further south in the Salish Sea, the patterns we're seeing are much more consistent with warm water, so that we don't see sea urchins in those places where we've lost kelp to a large degree anyway. But what we do see is that the places where kelp have been lost are places where the water has warmed up a lot, and up to 20 to 22 degrees Celsius in some places, which is beyond the physiological threshold that kelp can tolerate. And then the other thing you'll notice in this region is that there actually have been some gains. And so as you dig deeper into this, if you look at the sea otter counts for the coast, you'll realize that those are all places where sea otters are increasing exponentially since their reintroduction on the coast. And so sea otters also like sea urchins. And so as the sea otters are increasing, they're eating more sea urchins. That's removing those so that the kelp can then come back. So it seems like we're seeing those patterns there as well. Now, I do want to get to the Salish Sea, and I'll kind of work my way over there. So first, I'll just show you a quick slide from Barkley Sound, so the west coast of Vancouver Island. So we're kind of working our way there. And this is some of that first data that our students helped us collect back in 2016, and we've been monitoring these kelp forests from then till 2022. And if you take this one section of shoreline, you look from kind of the open ocean, open coast, where the water is quite cool, about 13 degrees in the summer or 14 degrees, up to the kind of further up into the sound where the water is at least 3 to 6 degrees warmer. So let's say 17, 18, even 20 degrees Celsius. We'll see that anything red is a place where kelp forests have been lost over the time that we've been monitoring. So you'll see that there are these pockets of warm water in some of these fjords and inlets like Barkley Sound, where the water is just getting too hot for the kelp forests at that point. And if we zoom in, and I guess if we look at the timing of that, we can also see that happened right around the same time the blob moved into our water. So there does seem to be a relationship between those warmer waters, the increased severity of these marine heat waves, and the loss of kelp forests. And then if you just quickly look into the Salish Sea region in here, as I mentioned, so we certainly see losses in that warmer kind of central and north Salish Sea region where there used to be kelp forests as late as 2007. They're much more rare in 2018 and onwards. That's a little bit true as you kind of work your way down into the Gulf Islands. And then as you kind of work your way into the Strait of Juan de Fuca, the kelp forests actually get quite stable. So these water patterns are very consistent with the kinds of losses we've seen. All right, so that's all the graphs I'm going to show you for tonight. And so, as we were discovering these patterns in BC, colleagues from Alaska to California have been seeing very similar patterns. And then, in lots of ways, scientists like myself and my colleagues have found ourselves kind of acting like coroners for these changes, right? We've been documenting these losses in great detail. We've been going to all these places and really showing the world what's happening. But, that in itself isn't actually, it's not doing anything about it. And so, I certainly have been, over the years asking myself, what's the value of this work? Why am I doing all this science? Why are we documenting these patterns? Because it's hard to see these losses even within my, relatively short lifetime. And so, we asked, what's the value? Like, why? If we don't do anything with this work, what's it for? And so I'm pleased to say that we did something. And so in 2021, I'd been doing this work for a while, mostly off the side of my desk, and I was approached by someone named John Page and his wife Goya Gann, who have lived in the Salish Sea for a long time, and had recently come into the position where they had some funds to put towards conservation issues, and they said, hey, you know what's happening with kelp forests, and what do we need to do to make a difference? Let's put some money and some effort towards this and see what we can do. And so we launched the Kelp Rescue Initiative in 2021 with this goal of using science to accelerate restoration of coastal ecosystems and to explore their potential to fight climate change. And we decided to focus on kind of 4 key regions. So one of them was Burrard Inlet. So that's that kind of urban kelp forest that I showed you in that initial photo. And in part, we did that because that's a place where we think we can learn a lot about why kelp forests are persisting and really use that to help us understand why they've disappeared from other places. We focused on the kind of North Salish Sea, so around Hornby Island and Denman Island and Comox, and all the way up to Quadra Island, in part because that's a place where there used to be those really abundant kelp forests, and they're in the 70s, let's say, and they're virtually gone now. So now the furthest south the kelp forest or the bull kelp forest extent into that part of the Salish Sea is Oyster River, so kind of halfway between Denman Island and Comox, or sorry, between Quadra Island and Comox, somewhere in there. The furthest north they extend, there used to be a bed right off the southern tip of Denman Island, and it was being studied and observed, and it blinked out in 2015. So that was as far north as they went in 2015. Now as far north as they go, certainly on the Vancouver Island side, is around the Nanaimo. And then we're working in Barkley Sound, where we've got a strong presence and a long history of work. And then up on the Broughton Archipelago, both because of its importance for salmon and salmon transitioning through that ecosystem, and because it has generally cooler waters. And so we got a whole bunch of scientists together and others, a whole bunch of partners, and said, let's see what we can do. What do we need to know? What are these gaps we need to fill to really understand why kelp forests are disappearing and how to restore them? And so this is everything from climate modelers who are building really detailed climate models for the Salish Sea and beyond, so we can really forecast what these places will look like in 2030 and 2050. It's genomics experts that are understanding how genetic variability of these different kelp forests vary between populations, so we can figure out which ones are losing genetic variability, which ones have important alleles or variants that might be useful in thermal performance, so in resistance to these heat waves. Community ecologists that are studying all the things that live in these kelp forests, and the goal really was to bring everybody together quickly and say, hey, this is urgent. We need to get as many people on board as possible to really do something. And so I'm really proud of the team we've assembled, and we've really been able to make some huge strides. And importantly, this work we're doing is very strongly connected to and supporting stewardship goals of First Nations with whom we work. And so that's true in all the regions where we're doing work. And so that would include the Comox First Nation in the Hornby and Denman Islands region and the North Salish Sea. And it's really to come to those nations and say, we've got this particular Western science knowledge, we've got this expertise. What are your goals, and what can we do to help? And so that's really been our focus. And so I'll spend the last part of my talk just telling you a little bit about the kinds of things that we can do to help kelp in these different places and where we are in that process. But to do that, I promised I wasn't going to show you any more graphs, and I won't, but I will show you one more diagram. So we need to take a little bit of a sort of nerdy dive into the life cycle of kelp forests or of kelp species, so we can understand how to use that to our advantage when we're thinking about restoration. So here's a picture of the kelp life cycle. And so what we think of when we think about kelp is usually this large, these canopy species. So bull kelp and giant kelp that form these big long fronds or blades that extend up to the surface. They've got these floats that carry them up. They can be 60 feet long in some places. And that's kind of, when we think about kelp, that's really all we think about. But that's only half of its life cycle. And so usually in the late summer, early fall, these kelp will they'll develop these spore patches. For giant kelp, it's on the base of their kind of near the kind of root-like structure near their holdfast. For bull kelp, it's along the length of their blades. I'll show you a picture in a sec. And those spore patches produce spores. Those spores actually swim around. They get released into the water. They swim around, they swim down, and they settle on the bottom, and they're microscopic. There's males and females. They grow into these little kind of mycelium-like structures that are growing along the bottom of the ocean. Can't see them. They're less than a millimeter in size, and they spend most of their dark winter down there. And at some point, males produce sperm. The sperm swims over and finds an egg that's on a female gametophyte. This microscopic stage, and that male sperm fertilizes the egg. It creates this microscopic embryo, and that embryo eventually in the next spring grows up into this giant 60-foot plant that you see. And so the reason I'm showing you this is because we can use this important pieces of the kelp life cycle to actually do something about these losses, to actually think about restoration, to actually seed new kelp in places where we think it's going to have the most value to salmon, or where we think it's going to be the most kind of resilient to the coming changes in climate that we see. And so here's just a couple more pictures. So this is a bull kelp from Alert Bay this fall. So just kind of laying along the beach here, you can see these blades extending out here off the end. And I pulled one of those off. And on it, you can see that dark patch in the middle. And so that whole dark patch there is all filled with these millions and millions of these microscopic spores. And so you can bring those spores into the lab. You can release them into a beaker, pour them into a petri dish, and grow them for a while. And after, say, a month, you'll end up with these little kind of fuzzy blobs. And if you put those under the microscope, they look like this. They're kind of this branching structure. And you can keep them in the lab for as long as you want, and you can even fragment them into smaller pieces, and they'll grow new blobs, and you can keep doing this over and over again until you have this whole, beaker or test tube full of these microscopic stages of kelp that are just ready to produce the next generation of these large adult kelps. So we do that. We bring them back to the lab. We culture them all winter. We set up aquarium tanks with small pebbles. In this case, we've got pebbles and tiles here. This is from some work that we did in Burrard Inlet. And you can actually seed those little microscopic gametophytes, those microscopic stages of kelp, right onto these rocks. You just kind of let them settle on, and they'll actually glue themselves onto there. And then this is just a zoomed in microscope image that you can see. You can see each one of these little kind of like fuzzy specks. That's a gametophyte. And each one of these little kind of leaf-like structures, that's a young sporophyte, so a young adult kelp that's only less than a millimeter long, that will, throughout the year grow up into these large individuals. If you keep them in the lab for a while, you can then turn them from microscopic into macroscopic sporophytes or adult kelp. So this is a piece of gravel that we're about to outplant near Hornby Island this year that has, let's say, 10 or 20 adult bull kelps growing on it. You can just barely see right here, there's little lines, the blades are just starting to split into an adult bull kelp. And so what you can do then is these are basically little kelp seeds that we've grown in the lab, and we can then go in a boat, we can release them off the side of the boat or take them down with divers and just plant them on the bottom of the ocean. And so this is each one of these little fuzzy patches here is a baby kelp sort of seed, a bunch of kelp put on a piece of gravel. We call this green gravel, even though it's brown, I guess. And then what happens is that the root-like structures, the holdfasts of these kelp, can actually grow off of the gravel, attached to the bedrock underneath, and then that will give them a firm anchor, so they can then send their floats and their leaves, their blades up to the surface to photosynthesize in the next generation. So this is just one of the tools that we're developing and that we're using, and this is something that's worked well everywhere from Norway to Australia to elsewhere. And it's certainly not perfect. We've found out this year as we've started piloting these projects in Barkley Sound and on Hornby and in Burrard Inlet that, a tray full of gravel with baby kelp on it is a lot heavier than a package of lettuce seeds, let's say, right, for your garden. And so sure, these are great in that you can scatter them from a boat in places where you want kelp to grow. You can grow them in the lab. We can make this happen. But it's not a technique that you can go out and reseed square kilometers of kelp forests without having a barge and some larger infrastructure. But we're excited to see the results so far, and it seems to be working quite well. And what you can see in the outline here is just a cage that we made, and this is work that we're doing in conjunction with Hornby Island Diving on a site called Mod Reef. And this cage is to keep the urchins out. And so the work we're doing right now there is really just to test, does this work for bull kelp? Does it work for giant kelp? Does it work for other species? And sort of what are the kind of limitations and what are the benefits and how long does it take, those kinds of things. So to do that, we've kept all the urchins at bay. But this is an image of a giant kelp forest off of Barkley Sound, and you can see, the healthy healthy giant kelp in the background, and you can see these sea urchins in the front. And so, any successful restoration approach, depending on what the main cause of the decline is in a different area. Is it warm water? Is it sedimentation? Is it sea urchin overgrazing? The kind of restoration solution of that site is going to vary. And so we're going to start some work next year actually looking at sea urchins, much like they've done folks from Parks Canada and the Haida Nation and others have done up in Haida Gwaii over a number of years to remove sea urchins from a stretch of shoreline. We'll do a bit of that work as well, just to assess different methods for removing sea urchins, assess the costs of those different methods so that we kind of have that as part of our toolkit as well. Now, the goal of all this work is to restore kelp forests in places where they have the most value. And so some of the other work that we're doing, and I mentioned this at the beginning, is in conjunction with climate modelers, for instance. So folks at University of Victoria, a scientist named Hansi Singh and some other folks working there in the climate modeling space are working to build some really high resolution, so very fine scale climate models, exactly the same ones that are used in the production of the IPCC reports, to create really fine scale and well-resolved climate models that work for nearshore places, that work for places where kelp forests live and work for places where eelgrass lives, and then use those to project those models into the future using these supercomputers and say, where are the places where those climate models say the water is just going to be too hot. Maybe it's not worth focusing a huge amount of our limited effort on those places. Maybe there's other things we can do in those sites to encourage productive habitat, maybe encourage forage fish spawning, other things, but it might not be a good place to think about seeding kelp because they're just going to get baked every year. Or some places where kelp might sort of be just hanging on in those future climate scenarios. So places where they might need some assisted evolution, so they might need some of the genomics work for, to figure out which lineages of kelp are likely to survive best there in the future. And then where are places where, it's likely to stay nice and cool. Kelp's a cold water species. They do well in cold water. So there might be a bunch of other sites in the Salish Sea, like around Victoria, for instance, or the Southern Gulf Islands, where, kelp are forecasted to do just fine in terms of water temperature, but we might want to focus on protecting them from other threats. So that might be some targeted urchin removals. That might be protecting them from harvesting or vessel traffic or anchoring of those other things. And then if we put all of that into the context of the places that we know salmon are migrating, or places where we think these kelp forest habitats are going to have the most value to salmon, to forage fish, to other species that depend on them, we might not be able to restore kelp forests, in every place in the Salish Sea where they've been lost. But if we can restore them in the places, we can use our limited effort to target the places with the most value to the species that depend on them, including people. Then the hope is that we can make a difference and move the needle in a positive direction. So I'll just end. This is all work that we're doing, and certainly, there's a bunch of us. There's First Nations communities involved. There's a bunch of Western scientists involved. But for people on this call, some people might ask, well, what can I do? What can I do to help kelp forests in the Salish Sea? If I've convinced you that they are indeed important, what can you do to help? Well, the first thing you can do, and I'll kind of end on the same slide that I started with, is to talk to your friends about kelp, talk to your elected officials about kelp forests, to really tell people about what you learned and something new you learned, why they're important. And then the other thing you can do, and again, this will depend on where you live in the Salish Sea, is go find your local kelp forest. There are these patches of kelp that are still persisting in a whole bunch of places. And I've put my email there, and I'm certainly happy for folks to reach out to me to say, hey, I live over here. Where's my nearest kelp forest? And I'll try to steer you in the right direction. Just to go both, watch it over the seasons, kind of be with it, see what you can see there, go at low tide, see if you can find some little baby kelp, in the intertidal zone. But also just have kind of some eyes on the water and eyes on those forests, so you can just keep track of how things are going there, and then reach out if things aren't looking good, or if they are. And certainly, if you enjoyed learning about this work, or you'd like to learn more about kelp forests or any of the work that we're doing, or any collaborators, please do visit our website at kelprescue.org. I put my email there. Feel free to reach out with questions. If I can't get to them, I'll try to refer you to some colleagues or collaborators that work with us. And then feel free to follow Kelp Rescue on Twitter and Instagram, and me at Neufeld5 as well.
I have had quite a few cups of coffee today, just a fair warning. Alrighty, so I'm so excited. Today I'm going to be talking about charismatic cephalopods of the Salish Sea. And for some of you who may be wondering, I went out 2 summers ago with my professor, Dr. Alan Verdi, to Rosario Beach Marine Lab in Anacortes, Washington, which is where I've gotten all of my knowledge on octopuses, especially those of the Salish Sea. So let me get my little laser pointer. So I'm over here, I'm in Maine. It's 11 p.m. right now, but I'm super excited to be teaching you guys about this. And then all the way over here was where I was. So 2 summers ago was my first time on the West Coast, and I am so excited to go back, hopefully one day. And then Chris's talk got me super excited, because last summer I was a scientific diving research intern at Bigelow Labs, where I worked with environmental DNA and also studied kelp forests in the Gulf of Maine. So there were a lot of parallels, but yeah. So kind of what my track is right now is we're gonna go over a little bit of like this taxonomy of octopuses, and then we'll move into some of the external and internal anatomy, and then we'll end on 3 big charismatic species of octopuses in the Salish Sea. But, all right, let's get it. How do I, boop. Okay, so mollusca is the taxonomic class that you find Cephalopoda in, but also as part of this phylum, in mollusca, there's also Polyplacophoria, which is chitin, so like the gumboot chitin. We have bivalvia, so clams, mussels, oysters, and scallops, and then also gastropoda, which are like your snails, your nudibranchs, abalone. But if we move from cephalopoda, which literally means head foot, because the members of this group have their head branch off into their arms, we get into octopoda, but there's also Sepioidea, which are cuttlefish, Teuthoidea, which are squids, and then Nautilida, which are nautiluses. And so something interesting to note is that octopuses evolved the most out of all the cephalopodas around 600 million years ago. And most of the cephalopods have retained some sort of shell as part of the mollusca class. But squids often have this little shell in them that's called a pen. It's their only thing that they retain, but octopuses do not have this at all. And then how I'm going to end this talk a little later on, I'm going to talk about the giant Pacific octopus, the ruby octopus, also known as the eastern red Pacific octopus, and then this little guy right here, who's the smooth skin octopus. All right, so let's start out with some external anatomy. So first, I'm going to talk about the mantle. And so right here, you can see this really good picture of a red that my professor actually took. The mantle is a highly muscled structure that houses in the internal organs of the octopus. And it uses the strong muscles of the mantle to basically protect the organs inside of it, like their gills and such. And it also contracts and relaxes to help with aid in respiration. And next, we'll move on to the head, which is like, just this little itty bitty part. And then their eyes, which are some of, one of the octopus's most amazing features other than their brain and nervous system. But so for their head, they have a central brain that's responsible for processing all of their visual information. And then we'll talk more about their brain later on. But as for the eyes, octopuses actually are colorblind, which most people would not think is what happens, but they actually have no cone photoreceptors for color, which is what we all have in our eyes. However, they do have these proteins called opsin that are light sensitive, and these actually help them detect light. And another interesting thing to note about octopuses is they have these charismatic rectangular shaped pupils that act as prisms. So basically what will happen is when the light hits their eye, it'll scatter the white light and it'll split them up into all these different color components. And so this effect is known as chromatic aberration. So that kind of leads into how they can actually detect the color and mimic their background environment. Also, another thing, interesting thing to note is that as octopuses change depth, their eyeball allows them to focus in on different wavelengths of light individually in the retina. So that is another interesting thing to think about. And it's not moving forward. There we go. Another thing that I just wanted to add on this slide is that octopuses have this other really unique, highly muscled system that basically they use to imitate the texture of surrounding objects. So as you can see, this rocky background, very bumpy, and they mimic very easily for a natural suit of camouflage using their circular and longitudinal muscles, so just how they contract and expand with a hydrostatic system. And these are called papillae, which, as you can see, helps them break up the outside of their environment. And moving on to the siphon. So the siphon is the tubular opening that you can see right here in the mantle. And this serves as a pathway for water in their environment. It can work as an expulsion of water, which allows for locomotion, like jet propulsion. You'll often see those videos underwater of like tropical octopuses where they'll be swimming and then they'll shoot water out and they go flying underwater. But it's also mainly used for excretion of wastes. And I have these photo or videos right here that should work. Yeah, so this is actually a video from the lab that I worked in. I was the lead aquarist that summer, so I did most of the octopus husbandry. And you can see right there, the siphon and the mantle are both contracting, and you can see the little jet stream of water. This octopus was a little disgruntled with me. He didn't really want to be messed with, but I was on a feeding schedule, so I had to help him out. And then here's another good one of these are both red octopuses, but he really gets me with his water siphon, and that's a purple shore crab that was one part of their diet that I was feeding them that summer. And another obviously important thing, charismatic thing to note about octopuses are their arms and their suckers. So they all, as we all know, octopus, they have 8 arms, and they each arm often has 2 rows of suckers, which they use for touch, smell, and taste. And something interesting to note is that 3/5 of the octopus's nervous system is distributed throughout their 8 arms. And so studies have found that essentially each of their individual arms has a mind of their own. And when they looked more into this, each arm has a little neural ring that can actually bypass the brain. So their arms are capable of passing messages along to one another without ever having to actually interact directly with the brain, the central brain, which, as I mentioned before, is mainly focusing on their visual aspects. So often you'll see them moving out and around and they're touching everything. They're getting all their information through their arms, in addition to what they're just seeing visually. And then another interesting thing that I'll mention briefly and get more to later is that male octopuses have something called a hectocotylus, which is a modified arm, which you can see in this moose octopus right here that I had that I needed to weigh. It's essentially a specialized arm that males use during reproduction to transfer their sperm packet to the female. And kind of looks like a worm. That's like what I always say. It looks like an earthworm, but basically that portion of the tentacle doesn't have any suckers along it anymore. And then this is some good visual on how their arms move. So as you can see, he's touching, he's tasting, he's smelling. I'll play that one again. But that's from the lab. And then this next video is actually from a GPO in a tire reef in Admiralty Bay, which is off of Whidbey Island. I don't know if anybody's familiar with that, but you can see their arm is moving, and I actually go in and put my glove up from my dry suit, and I'm like, hey there, little buddy. And yeah, you can see how strong their suckers are. But yeah, so chemoreceptors and lots of other touch, taste, smell aspects that they're using to explore their environment, which is very important for them as they are highly intelligent creatures. And so now I'm going to get a little more into the internal anatomy. So I'm going to talk about their beak and their poison gland. So all octopuses have this hard retractable beak that looks very similar to a parrot's beak, which you can see right here in the lower left-hand corner. And then I have this picture pointed how their mouth is actually central to their entire body. And so what they use their beak for is usually tearing open clamshells or other bivalves, and then they also use it to kind of tear apart the flesh that they're eating. And so this beak also has a little mouth component that's known as the radula, which is a barbed tongue that octopuses use to scrape the meat out of the shell once the shell of its prey is open. And snails and other mollusks also have this retained feature of the radula. So that's just kind of another thing to note. And then I have another video of this red octopus during feeding time that I'm giving a purple shore crab. And you'll see how it'll grab it and then it'll immediately envelop its prey. And what it does is it walks that purple shore crab using its suckers up to its mouth. And then it'll go, the video is a little bit longer, but it goes down to the bottom of its tank and it actually starts consuming the crab. But I can play that one again because I think it's really interesting to watch how they interact with everything. And look, you can see their arms also are just kind of all over the place looking around. And so this is some of my favorite studies that I've looked at about octopus, is that if their radula and their beak doesn't do the job for their food, they actually have another tooth-covered organ called the salivary papilla that it can use to actually drill holes into the shells. And basically what it does is that erodes the shell away. And this organ will also take toxins from its poison gland that it actually will use to paralyze its prey. And this can also be used as deterrence and defense against predators. And for when I first started working with the octopuses a couple summers ago, I didn't realize that they bit. I wasn't bit, but that's something to know when you're working. These are wild animals that we're using for, the purpose of research in science and conservation. But, it's important to respect them and make sure that they're okay in their new environment that's very unsettling potentially for them. But, sorry, some water. But, so this is actually, this diagram right here is from a study at the Seattle Aquarium that wanted to see how their red octopuses were drilling on these manila clams. So they wanted to see if there was a drill preference on this clam. And what they ended up finding is that there was a significant preference for drilling into the two adductor muscles, which are right here. So this is the anterior adductor muscle. This is the posterior adductor muscle. And basically what that muscle does is that's the one that actually keeps the shell clamped together. And so this is a strategic advantage for the octopuses, because if they drill right into that and then they inject their poison from their poison gland using the salivary papilla, and then it will cause the two shell halves to move apart. And so what they found is that about 80% of the drill holes were found over this anterior adductor muscle. And this is a little bit of an older study, but there's a lot of the mechanisms for actually figuring out how this works, how they know where to drill exactly is not necessarily known and is has more studies to figure it out. And then, so now I'm going to move on to the brain. So an octopus's brain is about proportionally as large as some birds and mammals' brains relative to their body size. So this actually displays a high level of organization in order to do things like coordinate all their chromatophoric color changes, which I'll get to in a little bit. And then octopuses, as many of you may have seen the film My Octopus Teacher, or even Finding Dory, that the octopus Hank was very, very intelligent. And in labs and just in general, they've been recorded to solve puzzles, escape from enclosures by memorizing guard schedules, as well as using and doing other cognitive tasks like using rocks as tools to get some of their prey open. And then another interesting thing is that they also have short and long-term memory, which has been demonstrated time and time again throughout behavioral studies. And a very recent study occurred in a lab in Japan, where they actually hooked up these electrodes to the octopuses to see their brain waves, and they just wanted to analyze their brain waves. And what they ended up seeing is they found a type of brain wave that they have never seen, but it was kind of similar to humans. So they're thinking that perhaps this may be a link to figuring out and finding out the origin or the evolution of intelligence. So more on that to come, hopefully, from that group. And so this is another interesting study that I just kind of wanted to include, a little complicated, but basically what these researchers did is they took a couple of different species of octopus, and their goal was to look at the central nervous system by looking at like the different foldings of their brain of a shallow tropical octopus species versus a deep sea species. And so they put them in an MRI machine and they analyzed the lobes of the brain. And essentially what they found is that the changes in the brain relative to the octopus is linked to their habits and their habitat. So if they're a diurnal octopus, their environment is more complex. There's more predators present, so they're going to have a more highly developed brain. Whereas if they're found in a less complex environment, there's going to be less predators, and they will not need to have as such demand for these highly intricate or significantly more intricate brain lobes. And so another thing to note is that octopuses have 3 hearts. So they have one systematic heart that pumps blood all throughout their body. And then they also have 2 brachial hearts, which actually pump blood through the gills, which is extremely important for them to gather oxygen from their environment and actually use it. And so this is a picture from the lab I worked in 2 summers ago. This arrow right here is pointing to a syringe, and as you can see, it looks blue. And so octopuses actually have blue blood, and this is due to copper being the metal that oxygen binds to, whereas in vertebrate blood, our blood is red because we have iron. And then one of the last things I'll talk about is the ctenidium. So the ctenidium is just a fancy word for gills. And so this is a really great picture, I thought, to add in there. So this is their mantle, there's the siphon, his little eyeball head. But right in there, this is a pretty good shot that shows their gills. And so just like a fish and other organisms, they extract oxygen from the water through this gill system, and then they excrete CO2 as a waste product. And then ink sacs. So an octopus can eject ink out of their sac along with a burst of water from the siphon. So this is often used in combination to fool predators. So they can create this like chic black cloud, or they can shoot out in little blobs like boop, 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 which can serve as decoys, or yeah. So basically it's, a defense mechanism. And interesting thing to add is that the ink of these octopuses contains alkaloids, often tyrosinase, which is a compound that impairs smell and taste. So once that ink is released into that area, anything in that surrounding area will face the effects, and it confuses the predator, and basically the chemoreceptors don't work properly in the predator. And an interesting thing to note is that their ink is actually toxic to them as well if they cannot escape the area, which is something very important to consider when you're looking at keeping these for scientific purposes. You need to make sure that you have a big enough environment. You're constantly checking that there's good water flow and everything like that. And so this I thought I muted that. But yeah, so you'll see this red is coming out. And so he grabs the crab, but check out all this color change that's going to be happening. And there he goes, right into his bottle. Oh, that's not what I want to do. But yeah, so I'll play that again. I'm going to play that again. There we go. Okay, so he's coming out of his bottle. He's looking. He sees the purple shore crab that I didn't even notice when I first saw this video. And then he envelops his prey. He's using all of his suckers and his arms to travel that up to his mouth, where he'll then use his beak to eat at the crab. And then at the same time, it's a good display on how color and texture changes they use. This is a little shout out to Dr. Onthank, Octopodium. He has a lot of great YouTube videos. He's the director of Rosario Marine Beach Lab. And so another, important anatomy thing to add is how octopuses actually can change their color. And so they do this through their chromatophores, and these are pigmented cells that basically have 3 little sacs of color in them, and there's tens of thousands of these that cover all of the octopus's skin. And fun fact, as I mentioned earlier, that octopuses have certain light reacting proteins in their eyes known as opsin. These molecules are also found in their chromatophores, which is distributed again throughout their skin. And so hypothetically, octopus can see, but they mostly just detect light with their skin. And so chromatophores are complex, atypically shaped cells, as you can see in this picture right here, super complicated. And basically, each chromatophore is surrounded by muscles, and that's what allows them to change how the pigment is being displayed by relaxing or contracting. And so each chromatophore is actually independently controlled by the nervous system, and this enables a high degree of complexity that can display these different colors. So when the muscle is relaxed, these pigments are often not seen, or they are a more neutral color. Whereas if the muscles are contracted, these pigments are highly visible. And there is a high level of control, as I just said, with the nervous system that allows them to change splashes across their skin in a flash, which you have seen in a few other videos, and I can always go back later if people want to see those again. But that's very important for their survival and using that as camouflage. And so this is just a little micrograph picture that's showing the muscular systems between these chromatophores, and then, the nervous system as well that's running along with these cells. So very complex system. And so, as I said, chromatophores are pigments, various colors, but there's also these really fun things called iridophores, also part of their skin, that reflect that light back at different angles. And this further allows octopuses to generate all these additional combination of colors that can accentuate all these dramatic changes by mirroring their surrounding environment. So they've really mastered it. Evolution has done them right for sure. And so kind of getting into the lifestyle of octopuses, like most creatures, the purpose of life is to reproduce. And then eventually these octopuses will succumb to their death. They only reproduce once. So what happens is a male will provide a female with a spermatophore with its hectocotylus that I pointed out earlier. And then she'll take that spermatophore and incorporate it with her eggs, and she'll wait for them to hatch. She won't get any food, and she'll stay with them in a den the entire time, which you can see in this bottle right here. That's a mother octopus, and those are her little eggs. And then here's another great picture of a female octopus with her babies in her den. And so what she'll do is she'll actually blow currents of water across the eggs, which keeps them clean with her siphon, and also protects them from like microscopic predators. And so, as I said, the female will pass away shortly after her eggs have hatched, and the male will pass away a few months afterward. And what we often see on the animal husbandry side is this natural process called senescence. And this often displays on octopuses as white lesions on their body, and they'll lessen their activity, and they also will often decrease their feeding as well. So more noticeable in males than females. And baby octopuses are very cute. So these are some little guys actually that came out of the mother's eggs. And they're so cute. They're so tiny, but you can also see their chromatophores are already active at even that small of size, which is smaller than a grain of rice. And more excitedly, we're moving into the actual species of octopus that we have, or you guys have in the Salish Sea. So I'm going to start out with the giant Pacific octopus. And so the giant Pacific octopus, as its name suggests, is the largest octopus species. It's usually reddish pink and it has a vein-like pattern. And they also have highly pronounced papillae. They like to make intricate patterns with their body that match their background. And they're found, they're found all around the Pacific. They've been found in Korea and Japan, as well as coastlines of Canada, Alaska, and the other portions of the Northwest Pacific on the United States. So they like cold water, that's for sure. They can also be found relatively shallow water. I've seen them as shallow as 20 feet in a bottle field, but I'll get on to that a little bit later. But they can also be found as deep as 1,500 meters. And octopuses, much all alike, they're relatively solitary animals, and they're often found alone in their den, except during mating season. And the giant Pacific octopus, believe it or not, has a very long lifespan. It's about 3 to 5 years. And that shocked me at first when I was first learning about octopuses. I had no idea how short their lives were, but just goes to show how cool critters they do, or how cool of critters they are in their short life they have. And so this is a giant Pacific octopus that I actually saw on a dive. So this was about 86 feet, which is, oh golly, I don't know how many meters that is. I'm not very good at math. Hopefully somebody can put that in the chat if they want to convert that, or if they know the conversion already. But yeah, so there's his eye. You can see the suckers on his arm. They were huge. That's a big boy or girl, all nestled up in their den. And so GPOs will often create these shell middens outside of their dens, and this is what is most famously referred to as octopus gardens. So as you can see, all this shell hash is actually a great indicator that there is an octopus nearby. We assume they make these dens outside of their homes because it's safer for them to eat inside their den rather than being out in the open where they're very vulnerable to predators. And again, as I just said, they're useful indicators for both divers, whether they be recreational or scientific, because this is a great indication that an octopus has lived here or it is currently living there. And we can kind of follow the gardens around. And they also provide really important insight to what they're eating. So as you can see, we got some manila clams. There's some scallop shells. This is some scallop shells from a dive that was right outside an octopus's den. Scallops, this one has a little sponge on it, but they're also really known to love Dungeness crabs, for sure. And so we're moving on to some predators of the giant Pacific octopus. You'd be like, what? Like, what's going to eat a giant octopus? Well, the answer is a lot of things. Some of the smaller ones, when they're juveniles, seabirds, if they're lucky enough, will eat octopus. Marine mammals like seals, otters, they're all known to eat octopus as well. And this is a lingcod, which some of you may be familiar with, but as you can see, that is a really great example of a predator in action. And so the next octopus is the East Pacific red octopus. I'll probably refer to them as red octos or rubies throughout the rest of the presentation. But this species lives in kelp forests with rocky or sandy bottoms. You can see this bottom right here is kind of sandy and muddy of the North Pacific, and they can be found to depths as deep as 200 meters. And where Rosario Lab has had their most success with finding these octos, it's been in Admiralty Bay, which is part of Whidbey Island, which is in the Salish Sea on the coast of Washington in Puget Sound. And something interesting about this is that they've actually made our trash their homes. So these octopus species have been very frequently seen living and denning in bottles, whether they be beer, wine. We've seen mason jars that they lived in, pickle jars, pretty much any glass vial, vessel, any glass vessel, they often will be found denning in. And so this is pretty unique and is an interesting way to represent how resilient these creatures have become to our pollution. And yeah, so this is actually from one of our density studies where we go to this bottle field and look for the octopus, and then we'll bring them back to the lab, record sex, weight, if there were eggs present in the bottle, all that jazz. And then we bring them back out and release them and hope that they reproduce and continue having a good life. But here's an example of one in the lab. As you can see, it has his own bottle. So we make sure that we keep them with their dens when we collect them, but then when we release them, they often will find new bottles to home in. And so this is actually a really unique picture because octopuses are not very social, except during mating season. So you can see all these reds are hanging out, getting ready to reproduce. And you can also note the color changes in this one. Definitely a lot spottier. It has some papillae breaking up, as does this one on its mantle. But very, very cool critters for sure. And then this is the smooth skin octopus. And this is my favorite octopus that I worked with the entire summer, Moose octopus leoderma. They are a very unique species of octopus because they're actually nocturnal, and they're a benthic species that are normally found in depths ranging from 250 to 1,400 meters in the North Pacific. So they are very deep, and at the lab I was working for is still not quite sure how they ended up getting where we were finding them. And so where we've had success finding these moose octopus is in Burroughs Bay, which is near Anacortes, and it's also near Rosario Marine Beach Lab. And so this study was actually done by a girl 2 years prior to me. So what she actually did was she was using one of those grab tools, and she was collecting sediment from the areas that they've been finding these moose octopus to see if she could figure out the isotopic components of different prey species. Because from what we've observed, they eat worms and small crustaceans and bivalves. And we've seen them as shallow as 9 meters in this area. So very different from their typical super deep sea location. And then this is super interesting and very unique to the species as well, is that they display a burrowing behavior in the sediment found in Burroughs Bay, which is a very uncommon behavior in cephalopods. And to date, the only other octopus known to display this burrowing behavior is octopus caruna, which is from Australia. So I'll play that again. You can see he's on top of the sediment right there, and then he slowly just wiggles his way in. And because this species is also nocturnal, how we find them, to collect them, to study them for lab, is we go out at night and we'll shine our flashlights, and while you're diving, swing it back and forth, and then you'll see them on the sediment. And most of the time they'll burrow, sometimes they escape, but occasionally you can get them. And this unique mantle flange right here that you can see on their mantle is thought to help with that burrowing behavior. A lot of other octopus species do not have this defined flange on their mantle. And those videos and other pictures may have been a little deceiving because they're actually super small. This is a little moose octopus on my hand. My hand's not very big, so about an inch maybe. Small size, like a golf ball or a head of garlic is what we usually say. But as you can see, there's that well-defined flange again, those alien-looking eyes, and then their relatively smooth appearance, which is pretty different from GPOs and the red octopus that we were seeing a little bit earlier. And so again, these enlarged alien eyes, which are part of their adaptation to that nocturnal activity, they display, as well as the deep sea lifestyle that they have. And they're also monochromatic in color, so they don't really have an ability to change their color, pattern, or even the texture of their skin through papillae, is one thing we've noticed. So as you can see, they're really usually this kind of orangey reddish color, very smooth appearance, and they have like very, very thin connective tissue that connects all their arms. And one other thing to note is that they have these little thin spindly arms. So their arms are relatively weak. In the GPOs, we saw how big those arms were. The suckers, they can be like, up to the size of like $50 or a quarter. That's US currency, but you get the point. Pretty big coins for how big their suckers are. Their arms, they don't really seem to have that much strength. So it's kind of a wonder what they do, what purpose they serve if they burrow in the deep, we're not really sure. But part of what the lab I was working for that was hoping to find is to create casts of their burrows so that we can understand how they aren't creating an anoxic environment when they actually burrow. If they burrow for a long time, there's a lot of unanswered questions that we're not really sure what to think about. But another thing, where's my laser? There it is. You can see those chromatophore cells really well in this picture right here, but they don't really seem to be doing much. They're just kind of creating this, nice color that is associated with the moose octopus in general. But yeah, so anatomy, internal and external, talked a little bit about taxonomic classes. And then we had those 3 species of octopus. We talked about GPOs, the red octopus, and then also the moose octopus, which is probably unique to some people because most people don't know that moose octopus are found in this area. But yeah, so thank you. Really appreciate it.
Thanks so much, Phoebe. That was super interesting. And I see some questions in the chat that Anne's gonna, I think, capture. And yeah, thanks so much.
Thank you.
Yeah, that was just great. Fascinating. Who knew? Honestly, we're all stuck on the great giant, and all these other ones are so amazing. All right, let's see. We have a question. How large are the chromatophores? I guess from Catherine, that would apply perhaps, well, shall we say the giant Pacific? How large would a chromatophore be measured across?
That's a good question. I'd say probably, when they expand and they contract with the muscles and with the muscle structure, they could probably be as big as half a centimeter, a couple of millimeters across, but they are just cells. So relatively small in comparison, but GPOs definitely have a lot more than some of the other species to cover their whole body surface area.
Yeah, exactly. And the moose would obviously have very tiny ones. So.
Yes, super.
Cool. Yeah. Okay, and Judy wonders, do they make more ink once expressed, or do they have only like a limited amount for lifetime? And does the ink kill other fish and plants as well as its predator?
From what I know, they are able to, it's not just like a one lifetime supply of ink. They're able to create more ink. And as for killing fish, I would not say, unless if maybe it's a really small larvae, that would impact too much, unless if they were in like an enclosed encounter. But in the ocean, there's a lot of current and direction that this travels. Some fish that might be in the area may be a little stunned and they might be a little confused. They might be like, hey, what's going on? But it's not really going to necessarily cause them to die.
Cool. All right. Do you ever find the little moose in plastic bottles, or do they prefer glass, or have you even studied that?
They definitely, from what the lab I worked at recorded, is they definitely prefer to den in glass bottles, or at least that area that they've been having success in finding them. It's typically glass bottles. It would be interesting to see otherwise. We did put out these PVC pipe, like little potential dens, but I don't think we ever looked at them while I was still there, but I know they're still out there. But yeah.
Cool. Well, amazing that we have so many bottles in the ocean, but anyway, whatever. All right. And what's the survival rate of the octopus larvae, for instance? That looked like a lot of eggs. What would be the % survival of all those?
Yeah, it's not very high. I think I read a statistic that was like 1 in every like 1,000 or to 10,000 actually survive and make it to adulthood. But that's how a lot of invertebrates are. They often do a lot of eggs in hopes that, you know, quite a few will survive or at least make it to the older juvenile state.
Yeah, exactly. And that sort of goes hand in hand with, you know, compared to an elephant that cares for one young for a long time, the octopus just let them go and that's it.
Yeah.
Not a lot of parental care in that. All right. And do they drill into clams by sort of scraping with that radula, or does that drill rotate in?
It kind of rotates in. So the radula is kind of like their little barbed tongue that they use to scrape out like the muscle mass from inside bivalves or crabs, what have you. But that other gland is called the salivary papilla, which is actually acts as a little drill that kind of erodes away at in combination usually with that toxin that they use at the shell. So it's kind of more, I'd say it's more of a drill.
More of a drill, huh? And it must take a little while, no? How long does
Yeah,
it take?
yeah, I know. Not a very like quick process, but
No, you're waiting for your dinner for quite some time, going away, drilling and scraping. Fascinating. So once a female lays the eggs, how long does it take for them to hatch? How long a gestation, I guess you might call it?
the gestation period is usually a few months. I'm pretty sure, I know GPOs can be in their dens with their eggs for a pretty long time, like 3 to 6 months, possibly even longer, depending on how it's all going.
Wow. That, like you say, then they expire after and they've hardly eaten or, right? That they
Yeah,
feed themselves or
right.
anything while they're doing that. Oh my gosh, I know that is really tough. And okay, well, I think that's most of the questions. I was just wondering myself, how are populations doing? Do you know anything much about the ecological?
I do not. I think I'm going to assume some of them, they're doing okay.
Right.
I haven't heard anything recently about their populations, you know, not doing super well, but you know, you never know. We have a lot of changing environments right now. You know, Chris noted the prolonged heat waves, those impacted invertebrates as well. So we just kind of got to keep on our toes.
Yes, for sure. I wonder if there's iNaturalist or anything. Can we report, like, if you are a swimmer or diver? I think there probably is. You know, you could probably if you see one, you could put it in there, and then sort of citizen science be able to try and get an idea of the population, perhaps. Very cool.